Good morning. Very welcome at this AMCRA event, the annual event where we present the results of the antimicrobial usage and antimicrobial resistance data. And this year we do it in combination with the scientific symposium. So in the morning session we'll have the scientists talking and we have plenty of interesting scientific presentations. And in the afternoon session we focus more on the results on the antimicrobial usage, the new data on usage and resistance of the previous year. So it's my great pleasure and honor to welcome you and to be the moderator of this morning session. And just to start off, it was a great success in terms of response we received. We received no less than 23 abstracts for this symposium. And not only from scientists in Belgium, but also from other countries such as Germany and Hungary. So it's really great to see that our symposium has some good attraction and a good follow-up by the scientists. So the symposium can be followed live as you are doing, but definitely also on the webinar. So many people are watching at home or from the office to this symposium. So also very welcome to the people following from a distance. And also this symposium will be recorded and later on will be put on the AMCRA website. So those of you that want to follow a presentation or have a look again to a certain presentation to go more in details, that is absolutely possible. The same is true for the posters. There are several posters outside in the hallway which you can look at during the coffee breaks, but the posters are also online available at the AMCRA website where you can read through them at your own pace. After each presentation, there will be room for questions from the audience here live present, but equally questions from the people at home. So those of you following at a distance, if you have a question, you can put them in the chat. Benedict here will follow up on the chat and will also read out your questions. So in that way, these questions are definitely also addressed. And with this practical information, it is my honor to introduce to you the first speaker. It's our invited keynote speaker, Dr. Els Broens from Utrecht University. Els was trained as a veterinarian at the Faculty of Veterinary Medicine of Utrecht, and she finished a PhD on livestock-associated MRSA in pigs. And since 2011, she's heading the Veterinary Microbiological Diagnostic Center at the Utrecht University. Els is also a diplomat in the European College of Veterinary Microbiology, and she's co-chairing the Cost Action Innovat, which is a very important cost action, working on diagnostics for antimicrobial resistance and good ways to monitor the resistance. Els, her research interests focus on zoonotic diseases and antimicrobial stewardship in veterinary medicine, and mainly the last topic is the one which is of interest for us today. And she has a very specific focus on this antimicrobial stewardship in companion animals, which is definitely going to be a topic for the coming years. So Els, we very much look forward to your presentation.
Thank you, Jeroen, and the organization for inviting me here. And I'm very happy to talk about, well, what my interest and my enthusiasm is about antimicrobial stewardship in companion animals. And maybe Jeroen said this will a topic for the next coming years, but maybe you're not all already convinced of that. And I will try to convince you that it is important. So I will talk about why, and then also about what and how. So first, why should we focus on companion animals? I think there are the main reasons I listed here is that the reasons why. First of all, we have the regulation, the EU regulation, which was implemented in 2022, which is not only applicable to farm animals, but also to our companion animals. So far, we had to focus on farm animals, and there is very limited data for companion animals, and also there's close contact. But I will go into these topics a little bit more in detail. First of all, the regulation for the EU, which was implemented last year. This is a legislation, this is applicable for all animal species, so not only for farm animals. And for instance, in our country, all kind of regulation and guidelines that we have, or the categorization for antibiotics, which is international, it's not only applicable for farm animals. So the the red or the third choice or the critical antimicrobials, these are just as critical for use in companion animals as in farm animals. And also where we now in a lot of countries already are monitoring use of antibiotics for farm animals, this has to be provided for all EU countries in 2024 for all farm animals. Well, of course, AMCRA and also in the Netherlands, we already do this. But in 2027, there should be more animals included in this monitoring. So all other food-producing animals, sheep, goat, and in 2030, dogs and cats data should be provided. So even if we don't think it is, we should because of this legislation. And so far there was this focus. There's only one slide in my presentation on farm animals, and this is just because I think maybe there will be more talks, and this afternoon we will see the new figures. But so far there has been focus on food-producing animals, and the data and results are very successful so far. So there's the EU Society for Veterinary Antimicrobial Consumption, the SVAC, which monitors this use for all for 31 countries, and you see there's a bit in a decline from 2011 to 2021 from 46.5%. In the Netherlands, we have the SDA, which does that already since 2009, and just the report came out last week, and we see a 70.4 decrease, and in Belgium, and this should be 2011 to 2020, there's also a very good decline. So there are successes by monitoring and all kind of actions we have implemented in farm animals. So, but there's very limited data in companion animals. And first of all, how do we quantify it? Of course, you can say, I can copy what I do in farm animals, but the data is not that good organized in companion animals as it is in the food-producing animals. So you can quantify in different ways. And if you look now in literature, different kind of quantifications are used, like total grams of active substances, which is of course what we use for food-producing animals as well, but also number of packages or number of tablets sold, and then implying that tablets are only used in companion animals, or number of prescriptions, number of treatments. But maybe the ideal thing is to work towards defined daily dose animals, just as we do in farm animals, so that we can a little bit compare this. Of course, that's hard to, what is the numerator for dogs and cats? But I think if we try to do that, we can maybe compare it a little bit more with what we do in the food-producing animals. So if you look at defined daily dose animal, then a number of 2 implies that the average dog, cat, and rabbit of this clinic, of a clinic, has received 2 days of antibiotic treatment in a year. So not all have had 2 days, but like one dog had for 3 weeks, and a lot of dogs didn't have any antibiotics at all, but on average 2 days a year. So SVAC, so the European Society has some data, but it's not very specified data on the use in antimicrobial use in companion animals, because they split the kilograms active substances in tablets, implying that these are used in companion animals, and the rest. So, and if you then look at the overall sales split by tablets, mainly used in companion animals versus all other products in 2021, you see for the total EU, for these 31 countries, 1.4% of the use is tablets. And I just highlighted out, Belgium is also 1.4%, in the Netherlands it's 2.2%. So it's low compared to in kilograms, but maybe we should look at what we are using in companion animals. Well, then there's some data, but this was only project-based from the Netherlands from 2009 till 2014. So after that, no data is available. So this is Dutch data where we had 100 veterinary practices, asking them to send us their prescription data. And on average in 2014, the DDDA, so the defined daily dose animal, was 2.6. But there was a large difference between practices. So because the range was very large, from 0.65 to 8.65. So it is very large differences. And you also see that the second choice antibiotics, which is the yellow, in the yellow bar, in the bars, the yellow is very high use of second choice antibiotics. And this is second choice for the Dutch categorization, because we have all kind of similar categorizations, but there are some differences between countries so far, and also differences with the EMA categorization, which has been published a few years ago. In the Netherlands, these are amoxicillin with clavulanic acid, and first-generation cephalosporins, but you see that there's a high use of these substances in companion animals. And the third choice antibiotics, which is in the Netherlands, is fluoroquinolones, and third and fourth generation cephalosporins is about 7%. So compared to what you see in food-producing animals, second and third choice use is rather high. Then in Denmark, they do a really kind of structural registration for antibiotics in dogs and, pets and horses. So, but it's still an estimation because, again, they look at tablets and to products that are only registered for these animals, but it's still, I think, a sort of estimation, but it is a good guess, I guess. So the estimated antibiotic use for horses and pets in kilograms in Denmark for 2021, well, you see in the graph that's from 2012 till 2021. The orange line is what is in horses, so only medicine registered for horses. The blue line is medicine only registered for dogs and cats. And the gray one is unspecified, but most probably used in horses or pets. So, and you see there is an 8% increase of use between 2017 and 2021. So instead of the decrease we see in the food-producing animals, we see an increase here. A large proportion for dogs and cats we know is used for chronic or recurrent disease, for instance, pyoderma or otitis externa. And the use of fluoroquinolones in companion animals in Denmark in 2021, 90% of all fluoroquinolones used in animals. So the fluoroquinolone use in food production animals is very low, but compared in pets, it's quite high. And the use of cephalosporins in companion animals is about 60% of all cephalosporins in animals in Denmark. And if you look at even third and fourth generation cephalosporins, that was only used in companion animals. So no use in food-producing animals, but only in companion animals. So summarizing this part, limited data. So there's no central registration and quantification of AMU. Instead of maybe in Denmark, they do a very good thing there, but still it's an estimation. But in all other countries, it's project-based most of the time, and only like for a few years. The total use of AMU is low compared to food-producing animals. As I said, 1.4% of total use in kilograms. But you see no or a small, even an increase or a very small decline in AMU compared to food-producing animals. And the use of critically important antimicrobials, including those registered for human use only, is relatively high in companion animals. So the focus in companion animals should not be on quality and appropriateness more than on quantity, maybe. So also we have to realize that the contact between humans and pets is much higher than between food-producing animals and humans. Of course, a farmer will have close contact with his animals, but this is only one farmer and people working in the food-producing industry. But like 50% of households have a dog or cat, meaning that all these people living in this household have most of the time close contact with these pets. And Philip Joosten here from Belgium did a nice study where he asked owners how intense contact they had, and almost all indicated there was at least one of the intense interactions. For instance, licking a hand or face. And about 1/3 of the dogs slept in the owner's bed. I don't think it's very hygienic, but I know it happens. My mother does it all the time. I tell her not to, but she does. So there's definitely close contact. And we have to realize that animals carry resistant bacteria. If you look in literature, resistant bacteria are widespread in companion animals. Prevalences vary a lot, and also study designs varies a lot, so it's hard to really set prevalences. But there are several publications, and I just highlighted here some, but on MRSA transmission from dogs to humans, MRSP transmission, so the pseudointermediates, which is methicillin-resistant staphylococcus, from dogs, but also you see that there is transmission to owners or veterinarians. ESBL, so the extended spectrum beta-lactamases, also we've seen them in dogs and cats and transmission within the household. And even the carbapenem resistance, we see that in dogs, and I think there's a talk about that just after me. So we have to be aware that there's close contact and that transmission occurs. So I looked into the website of AMCRA to see what is happening on companion animal side, and I looked at the action points and I just now highlighted the ones focusing on companion animals. And so the plans are to establish a system for monitoring antibiotic use in companion animals, which should be operational by 2024. So I hope that that you will be lucky to do that, because I think, well, of course, we have time till 2030, but it would be good if we do that as soon as possible. And also, it's benchmarking of veterinarians in non-food producing animal sector must report their antibiotic use data, so that there can be benchmarking from these veterinarians. And here, the third point is on antibiotics only authorized for human use. So monitoring the legitimacy, because what you've seen from Denmark is that these are only used in companion animals. And also the red list antibiotics should be, so the regulations should be extended to all species, and also monitoring of antibiotic resistance should be extended to non-food producing species. So it's not only me thinking that companion animals should be focused on, but I think AMCA will do that the coming years as well. So I hope I'm convinced you that we should focus on companion animals, but then how, or what and how are we doing this? So first of all, maybe for those who are not familiar with the term antimicrobial stewardship, I would like to share some definitions, because there are multiple definitions of antimicrobial stewardship. And I just listed here 3. And the first one is from 2007. They are all from the human side, because in human medicine, antimicrobial stewardship is very already wide out spread and very successful. So the primary goal of antimicrobial stewardship is to optimize clinical patient outcomes while minimizing unintended consequences of antimicrobial use, including the emergence of AMR in this case. Okay, and then another definition is a coherent set of actions. So remember, a set of actions, it's not just one button to push, which promote using antimicrobials responsibly. And another one is the persistent effort. And this is, of course, also important. It's just not one click and then it's done. You have to keep trying and doing things. So it's a persistent effort by a healthcare institute, which could be a veterinary practice or a veterinarian on his own, to measure and improve the appropriate use of antimicrobial agents. And maybe to summarize, as little as possible and only as much as necessary. So I'm not pleading to go to 0, not at all, because we need antibiotics, but we should use them appropriate. And this is a figure just to show you that veterinary antimicrobial stewardship is not just one thing, it's a set of things, and you don't have to apply them all at once, but you can pick where you think the most action is needed, or maybe the best success is expected. And in this paper from Prescott, he also says it's the 5 R's, which is responsibility, reduction, replacement, refinement, and review of the last is evaluate if your treatment that you prescribed, whether it worked, yes or no. Another thing is here the building blocks for a successful antimicrobial stewardship program. And again, this is from human medicine, meaning I did a course 2 weeks ago on antibiotic stewardship in human medicine, and I was first very inspired, but also a bit frustrated, because a lot of the prerequisites, which I just highlighted here on the left side of the pyramid, like a stewardship team, financial support, and guidelines, are things we are most time lacking in veterinary medicine. So, and stewardship team, I think that's something for a hospital, but not for a private practice maybe. But financial support, we need financial support, and we need also guidelines. And in this NOVAT, Jeroen already told this, and I will talk a little bit more about this. So this cost action, this network for European treatment optimization, there was this paper on an overview and evaluation of existing guidelines for rational antimicrobial use in small animal veterinary practice in Europe. I know the AMCRA guideline was in there, the Dutch guideline was in there. So we asked 40 countries, and 11 countries had a guideline, which was reviewed. In total, there were 15 guidelines because there were 4 more like international guidelines included as well. And the main conclusion was that the recommendations are often based on expert opinion and extrapolation from human medicine because clinical data is lacking. There's no transparency on the decision process, so it could be that this very stubborn professor was just pushing all his opinions, could be, or it could be a very democratic process, but there's no transparency. There's also no input or feedback from stakeholders, for instance, pet owners, and also there's no data on implementation and adherence of guidelines. So even if you have this guideline, it could be somewhere on a website or in a bookshelf, but if nobody's using it, there's no data on that, on how people, if and how people implement guidelines. So there's a need for evidence-based antimicrobial treatment guidelines, not only in companion animals, but of course I'm talking about companion animals today, and ENOVAT is working on that. They're working on 2 ENOVAT guidelines for companion animals, one on canine acute diarrhea, I think a indication where we misuse or overuse antibiotics a lot, and the same counts for surgical prophylaxis, and the publication of these guidelines is planned for 2024. So these are European or international guidelines. Might be that you have to apply them a little bit to your own national situation, but these are, I think these are done with a very rigorous process of reviewing and transparency. So okay, that was the what or the how. So I want just to present a Dutch example, which was a project-based. So we did a project which was financed, and so we didn't succeed in continuing this, but it was a quite very good and nice project, where we had this we called it the ASAP project, antimicrobial stewardship and pets, and the goal was to develop, implement, and evaluate the effectiveness of antimicrobial stewardship program in companion animal practices, and the sub-goals were to increase awareness on antibiotic use, to decrease total antibiotic use whenever possible, and also to shift antibiotic use towards from second and third choice to first choice antibiotics. So what we did is first we interviewed a set of veterinarians on how they prescribe antibiotics and what are the barriers or facilitators to do it right, yes or no, and if they know the guidelines, if they use the guidelines and things like that. We also did a survey and we looked into literature and we looked at the guidelines we have in the Netherlands for companion animals and expert opinion, and based on all these data we developed a stewardship program, which was a set of actions, so a multifaceted approach, as I said in one of the definitions already. So we did education. We educated the veterinarians on antibiotic resistance and antibiotic use and on the regulations in place, and also about the guidelines that were in place at that time. We quantified and benchmarked the antibiotic use of the clinics that were included. We gave the veterinarians individual feedback on prescription. We asked them to fill in forms for 100 patients they had on what they did. And then we analyzed that and went to visit the practice to give individual feedback on what is going well, where to improve and whatever. So there was individual feedback. And we also developed an information leaflet for animal owners, which they could give to their owners to say, like, I think there are human examples where you say flu, we don't need antibiotics for flu, and also to inform animal owners on antibiotic resistance and things like that. So, and I think these are the main buttons to push. First of all, there is this patient with, for instance, diarrhea. Does it need antibiotic, yes or no? If you say no, that's good. If you say yes, which antibiotic am I going to give? Do I going to give a first choice, a narrow spectrum one, or a broad spectrum one, or maybe even fluoroquinolones? So think of that. So these are the think moments in your process of decision making as a veterinarian where you can push a button. And then if you decided to give a antibiotic, for how long? I think we are, well, I'm already a veterinarian for 25 years, I think. So I know like 20 years ago, we said, well, let's do 10 days. And now, if you only reduce that to 5 days, for many of the indications or many prescriptions you do, you reduce your antibiotic use with 50%. And most of the time, 5 days is long enough. Of course, it depends on the indication, but think of, do we really need 10 days? Do we really need 2 weeks? Or like in just a simple cystitis, we're going shorter and shorter. In human medicine, it's sometimes just 1 day. Why should it be a week in dogs? Of course, sometimes evidence is lacking, but maybe try it sometimes. And make a call to the owner, like, how is he going? Should we prolong, yes or no, instead of giving tablets for 10 days. So what we do, we had 44 companion animal clinics, and we asked them to get their baseline data before we came, and then we implemented the program, and then we looked at the antibiotic use afterwards. And these are the baseline data. And again, green is first choice antibiotics, yellow is second choice antibiotics, and third choice is red. And this is according to the Dutch guidelines. And you see from 2012 to 2015 that there is a decrease in antibiotic use in total from 1.82 to 1.56. And you see a shift towards first choice. Well, you see the green bars are about the same, where the total decreases. And second choice is still most used, as I showed you before. And third choice is decreasing. So the red bars are decreasing. So this is before we came. So this is data, retrospective data before we came. Then we implemented the stewardship program. And again, we looked at the data in the years and the period after that. And we saw on top of the ongoing decreasing trend, you just saw that there was, again, there was a significant decrease of 15% in total antibiotic use, and also that the slope was steeper. So it was, if there was a decrease already, then we saw that the slope was going down even more steep. And this was for first choice antibiotics, even bigger decrease of 26% for second choice antibiotics. We didn't see a significant decrease in third choice, but that was because third choice was already very low, so it's very hard to see a significant effect. And we saw like it was totally dispersed per clinic. There was a lot of variation, because you can imagine if you already are very low and you have one patient that you have to treat for 2 weeks with fluoroquinolones, your figures go up sky high. So that's why there was no significant decrease there. So conclusions of our project, what we did, is that the baseline data before we came already showed a decrease in total AMU and a shift towards first choice antibiotics already, probably due to that we all had all kind of action in the Netherlands already on food producing animals. So also the companion animal veterinarians were like, maybe we should do something. Then we implemented the program and we saw an even further reduction in optimization of antibiotic use. And in the interviews we did afterwards, the participants indicated that they had an increased awareness of antibiotic use, so they were more aware of what they were doing in this decision process, yes or no, which antibiotic, how long. And what they said is the individual feedback and advice appeared to be very effective. They said that was the most, because we really said, well, you are using this, maybe you can do this better, or you can do this. And that was very effective, but also very time consuming. And as I said before I presented this, it's very hard to continue this kind of intensive programming for a long time. So there are other examples, and I just want to show you one other example from Australia, where they had an antimicrobial stewardship program, and they had 3 levels. So very low-key, they only educated the veterinarians. Then they had the intermediate intervention, where they also had more guidelines, and they also had like the green, yellow, red, which is shown here, and for the people looking online, so the circles on the right side of the screen, green, so the first choice, second choice, third choice antibiotics. And then there was a very intensive intervention where there was more feedback and things like that. So And what they saw that in all 3 levels there was a reduction of antibiotic use and also a shift towards prescribing low importance antibiotics. There was a greater reduction achieved with intensive program than with the less intensive programs, and the greatest impact was seen in the top 25% high prescribers. So antimicrobial stewardship is, well, actually what we try to do is influence behavior of people, which is a science on its own. So we are veterinarians and we know about infectious diseases and antibiotics, but we don't know anything really about behavior. Well, I think the average vet is very stubborn, so it might be even harder to influence behavior of veterinarians, but we need people from these kind of science to influence behavior, and we have to realize that it's influenced by many factors. Are you in a hurry? Are you stressed? Is there an owner very demanding? Things like that. So we need a set of actions, a bundled interventions attuned to the specific setting and influencing factors AMU, including quantification, education, guidelines, incentives, dedicated people, and of course time and money. So just here, there is no magic bullet. You have to look at your situation. So just to summarize, antimicrobial stewardship in companion animals, why, what and how. So why? Because there's this legislation. We have limited data, but we have very close contact with our pets, and transmission of resistant bacteria occurs. What? We need guidelines and we need quantification of antibiotic use at least. And how? By a multifaceted antimicrobial stewardship program. So that's what I want to say this morning. Thanks for listening. Enjoy the rest of the day. And if there are any questions, I'm happy to answer.
Thank you very much, Els, for this very interesting and inspiring presentation. Are there any questions from the audience here live? I see a hand raised in the back. Somebody will come to you with the microphone.
Thank you for this amazing talk. You showed some data about Denmark, and you said that the most antimicrobials are used for recurrent diseases like pyoderma and otitis externa. I was wondering if there is such data available in the Netherlands or in Belgium.
Well, I'm not sure about Belgium, but in the Netherlands, no, it's not. Even in food-producing animals, we still don't know where the antibiotics, for which indication they are used. So, and for companion animals, that's even farther away. Of course, that would be brilliant to have that. And for in like small projects, we probably know, but not for all use. And I guess in Belgium, it's the same. But
it's the same.
Yeah.
The data we have is based on small studies, but there is no general data on this. No.
And would you say it's comparable? That the situation in Denmark is comparable to the Netherlands and Belgium?
Yes. Yes.
Thanks for the very nice presentation. I was curious about the incentives that you used in your program in the Netherlands for the companion animal practice.
Well, what we did in a project is that the practices got some small amount of money. So it was not that they just did it because they got thousands of euros, but just like a little compensation of their time. But asking them afterwards, they said, well, I would have even done it without the financial compensation, but realized it's only 44 practices, which were very enthusiastic about the topic already. So it will be hard to also include all practices and the people who are less interested in this topic. So not sure, maybe, because of course in the Netherlands, if you look at the food-producing animals, I think there were not maybe an incentive, but there were fines and punishment if you did it wrong. Not sure if that is, but I think you have to have a balance there.
Okay, I see there is a question here from the online audience. Benedict will say it. Yep.
You hear me? Yeah. I have several questions in the chat. The first question is from Hanne Debergh. She asked for some indications. Scientific information is indeed available that the duration that an antibiotic has to be given can be decreased. However, according to the EU legislation, veterinarians are not allowed to deviate from the dose and duration put down in the SPC, according to an article of the EU legislation. How do you deal with this in the Netherlands and in Belgium?
Okay, so in the Netherlands, what we do is we say shortening of duration. We have this general guideline for all animals is that shortness of duration is always that you can do that without any problem. So that's in our guideline, and also our ministry, our government said, okay, that's okay. If you want to prolong, of course, sometimes you have to prolong. That's more difficult, but shortening is no problem. That's what we put in the guidelines. But and then our government says that's okay. But I'm not sure about if you do this in Belgium the same way.
It's a good suggestion for our government, I think.
Then I have a question from Geert Hoflak. One-day treatment in human medicine is with concentration-dependent products like fluoroquinolones. Time-dependent antibiotics cannot be used for that. So we should be careful when suggesting to shorten therapy length without taking the molecule into account, he says.
I totally agree. So that's what we should be in our guidelines, because for some drugs you can shorten and for others you need at least 2, 3, 4, 5 days. Yeah.
Another question is from Dominique Janssens. He says there are guidelines on one side, which are based on active substance, and then we have the legislation, like the EU legislation from 2019, with cascade rules on the other side, which are based on indication and species. Both are often in conflict. How do we have to cope with this?
I think that's the big challenge for all countries to look at the legislation and the different guidelines that are out there, which are, of course, the legislation is not an advice, but there are guidelines from the EMA or whatever, which are advices that you have to implement in your own country. And I'm also already aware that different countries interpret even the legislation on a different way. So I think in contact with your government, and with the experts, of course, in the different authorities, like AMCRA and SDA and the WVAB in the Netherlands, you have to look how strict would we want to implement this legislation, and can we sometimes maybe deviate a little bit of course, in conversation with your government and with evidence based on science that shortening or maybe higher dose, because of course we have some very ancient products on the market with doses which are not very good anymore. So can you maybe deviate that? But I'm sorry, but I think we have to do that on a national level, and we can't do that on EU level yet.
And one final question, how is antimicrobial stewardship practically organized in the Netherlands? This question is from Philip Boyen.
Well, Philip, hello, Philip. As I said, it was in this project and it's not organized at all at the moment, unfortunately.
Okay, thank you. There are no further questions in the chat.
Okay, thank you very much. Thank you once again, Els, for this brilliant presentation and the good discussion we had afterwards. And with this, we go to our second presenter, and this is the first selected presentation, which is coming from Hanne Debergh from Sciensano. And she will present the first Belgian report on ertapenem resistance in an ST11 Klebsiella pneumonia strain isolated from a dog carrying BLA SCO1 and BLA DHA1 combined with permeability defects. Hanne, the word is yours.
Thank you. Yeah. Thank you. Okay, good morning, everyone. So my talk is a great continuation of the previous talk, and indeed it will be about ertapenem resistance in an ST11 Klebsiella pneumoniae strain that we isolated from a companion animal. So first, a very big, broad overview of the PhD I'm currently working on, which is is a goal-oriented research, which tackles the problem of certain gaps in the monitoring program of drug resistance in Belgium. And this problem we try to tackle with two model pathogens. So on the one hand, Klebsiella pneumoniae, and on the other hand, the fungus Aspergillus fumigatus. But I will not talk about that today. And we selected those 2 pathogens because none of them is included in the National AMR Monitoring Program. However, they do pose a great threat to public health in both humans and animals. And they can cause severe and life-threatening infections, as I will discuss later. They are resistant, or they can be resistant, to a number of critical important antimicrobials, and most of all, their occurrence of those resistance isolates is not mapped in the environment in Belgium. So we try to tackle this problem using a One Health approach, where we look at the interstition between animals, humans, and the environment. And in the end, the goal is to develop some guidelines. So Klebsiella pneumoniae is a gram-negative bacterium, which is common in the environment, as well as in human and animal microbiota. However, it can be an opportunistic pathogen causing pneumonia, bloodstream infections, wound infections, but also UTIs. As I said, they can present with antibiotic resistance, so many of them are ESBL producing or carbapenem resistant, which is also why they are taken up in the list of urgent threats by the WHO. Klebsiella has been called the canary in the coal mine also because some of the known resistance genes have been first detected in Klebsiella, and it's a known trafficker of resistance genes. So it takes up antibiotic resistance genes and plasmid really easily, but also traffics them really easily. So as we are in a One Health concept, this is a scheme showing that really there is all those environments are intertwined. So humans with their excretions coming into the sewage, effluent, then in the environment, and so on and so on. There's really, they really interplay all together. Today I will focus only on animals. And related to this, as discussed before, the EMA has some categorizations on antibiotics of use in animals to advise on prudent and responsible use in both animals but also humans. And one of those, and those categories can be divided in 4 categories. So the first one is the antibiotics that should be avoided. Then the second one is those that should be restricted. Then category C is caution, and the last category D is those that need to be used with prudence. So category A is the ones to avoid, and those are the ones that are not authorized in the EU as veterinary medicine, so they should not be used in food-producing animals. However, in certain circumstances, they can be used in companion animals. And if we look at those antibiotics included in that category, amongst others, of course, there are the carbapenems, so for to say meropenem, doripenem, and we will look more into those. So carbapenem resistance can occur using or via different mechanisms. The first one being porin mutations, and those prevent antibiotic entry into the cell. So the antibiotic concentration is not enough to have an effect. The second one are the carbapenemases. So there we have an enzymatic inactivation of the antibiotic, and there are also 4 types of carbapenemase classes. For example, in class A you have the KPC, which is plasmid encoded, which is quite frequent in Klebsiella. And then in class D you have the OXA, for example, OXA48, which causes also carbapenem resistance. And then a last resistance mechanism is the increased efflux of antibiotics, again decreasing the concentration in the cell. For the rest of my talk, I will discuss a case study on carbapenem resistance in a companion animal, more in detail, a 4-month-old female Rottweiler dog, which died in June 2020 of fatal sepsis caused by Klebsiella pneumoniae. Postmortem biopsies were performed on liver and brain. However, it would have been interesting for our case, but we did not receive any information on possible treatments, nor were we able to receive a sample from the owner due to privacy reasons. And so we investigated the resistance mechanism of this ertapenem-resistant Klebsiella pneumoniae strain coming from a companion animal, and this was also published in Antibiotics. So we started, or at least Arcia started with the dog that they received, where they performed postmortem analysis, biopsies of the brain and the liver. Then they streaked it onto Gassner medium and Columbia, Gassner for Enterobacteriaceae detection. Presumptive Klebsiella pneumoniae strains were then seeded onto nutrient agar, and then species identification was performed with MALDI-TOF MS. Of those Klebsiella pneumoniae strains, we performed antimicrobial sensitivity testing, which we started first with the first panel, but then we saw that there are some interesting resistance phenotypes, so we went on with the second panel to look more into detail for that. And there, I will show you in the next slide, we saw some very interesting resistance phenotypes, so we went on with Illumina sequencing, and then in a later stage also nanopore sequencing, so we could perform a hybrid sequencing to also circularize the plasmids. So this is the antibiotic resistance phenotype in panel 1 on the left and panel 2 on the right. And if we look at the antibiotics or where it was resistant to, it's quite a lot. So it was a heavy multi-drug resistant strain. So it had an ESBL and AMP-C phenotype, but then maybe more important for this talk is that we saw the ertapenem resistance. So in the right column, we had ertapenem resistance. However, we did not observe any resistance to imipenem or meropenem, which is quite surprising. And we also observed temocillin resistance. So as I said before, we went into sequencing, so hybrid sequencing, to look more into the mechanisms. So we observed that it was an ST11, which is it's called a global problem clone, as it is often MDR, so multi-drug resistant, and it's also a clinical strain, indicative of a transmission from dog to human, human to dog. But as I said, we did not get any isolate from the owner, so we cannot investigate it. But then, as I said, it's a global problem clone, so it's ST11 is part of clonal group 258, which you see in the box here on panel A. And if we look more into detail on panel B on the right, then you see on the bottom clonal group 258, which is often resistant to carbapenems, but also aminoglycosides and sulfonamides. And then if you look on the table on the left, which is a genotypic resistance of our strain, then you see that we observed 3 plasmids, so INC-F1b, INC-R, and col-RNA-A. INC-F1b carried several antibiotic resistance genes, for example, towards the beta-lactam, so the SCO1, SHV2. It also had resistance against the aminoglycosides, sulfonamides, and so on. So it really matches what we usually observe in that clonal group. However, as I said before, it has ertapenem resistance, but here we do not observe a carbapenemase gene. So we wanted to look more into detail into that, what could have caused this phenotype. So on the INC-F1b plasmid, we have the SCO1 gene, which has a weak carbapenemase activity. However, it doesn't, only that doesn't really describe the phenotype that we saw. So we went more into detail with the hybrid sequencing. So we performed circularization of the plasmids. That's to say, of the 2 plasmids carrying the antibiotic resistance genes, of the 3rd one that did not carry, we did not perform it. So in this figure, you can see our plasmid, which is the light blue one on the middle, and then the other 3 rings with the different colors, they represent other plasmids that are highly similar to the ones that we observed. However, you do observe that the MDR part, so the bottom part, with all the genes in yellow, those are the antibiotic resistance genes, they do not match with all the other plasmids. So this could mean that the MDR parts were received from other plasmids or from other bacteria. And this is also seen with all the insertion sequences in blue here on the plasmids. Then on the right, the bottom right, you see the SCO1 present on the plasmid. And another thing that is important to notice is here on the left that you see all the transfer genes, so meaning that this plasmid is conjugative, so it can be spread to other bacteria, which of course is a bit worrying. The second plasmid that we circularized was the INC-R plasmid carrying the DHA1 gene, so the AMP-C that led to the AMP-C phenotype. And here you see, of course, apart from the DHA1, some other resistance genes on the top in yellow. For example, the SOL1 for sulfonamides, the AAC, and so on for the aminoglycosides. But then, okay, we have the DHA1, we have the SCO1, but still, it's a bit difficult to really, say that this could cause ertapenem resistance. So we went to the chromosome, looking into the outer membrane porins. If there are some mutations present that could cause that phenotype. So we, in the OMPK37, as you recall a few slides back, we observed the OMPK35 and 36. But here we observed the OMPK37 bearing some mutations. So all in all, this has led to the ertapenem resistance in our case. So now we wonder, is this a single case, of course? So in my PhD, we have 139 Klebsiella pneumoniae strains with ertapenem resistance. And so if you follow the algorithm, we'll look into first the sensitive to meropenem, sensitive to imipenem, and then we come to 2 different types. So one resistant to temacillin, and then the one sensitive to temacillin. And there we look into the MC phenotype. We have 8 that are resistant to temacillin and 11 that are sensitive to temacillin. But this is an ongoing study to see if this really is a single case or if we can extrapolate it to other ertapenem resistance strains. So to conclude, we described here the fatal sepsis of a 4-month-old Rottweiler, which had an MC phenotype with ertapenem resistance, meropenem and imipenem sensitivity. No carbapenemase genes were found. However, we did describe 3 plasmids, one of them which was conjugative and had the SCO1 gene. And then together with the SCO1, the DHA1, and the outer membrane permeability defects, this has led to the ertapenem resistance, and further investigations are ongoing. So with that, I would like to thank you for your attention and the people who worked with this. For me, thank you.
Thank you very much for, again, a very interesting presentation. Are there any questions? I don't see any immediate questions. I'm also looking to the chat. No questions on the chat. Maybe I can ask you a question. In the meantime, people can think of others. Do you have any, is there any idea of setting up a kind of a surveillance system or monitoring system to catch more of these strains? Because they are hugely interesting, and you, I understand, you indicated the limitations that you have little information about the dog and the treatment history of the dog, but future, maybe in the future you could get that. Do you have any plans for that?
No, not really, but as there is going to be a surveillance on companion animals, it would be interesting to include also Klebsiella pneumoniae, as it really is an upcoming pathogen. So I think it's important to, especially as there's a very close contact with humans and their pets, I think it's important to include it.
Okay, thank you. Any more questions in the back, Bob?
Do you hear me? Okay. Yeah. Thank you for the lecture. I was just wondering if the fatal outcome of this Rottweiler was Klebsiella the primary cause or the secondary cause, or were there no other things involved, viral diseases, and Klebsiella was just the guy who benefited from the dog being ill?
No, it was really a fatal sepsis due to the Klebsiella. Yeah.
Thank you.
Thank you.
Okay, thank you once more. And with this, we can go to our second selected presentation, and this one is coming from Evelien Biebaut from our own university, Gent University. And Evelien will report on the results of a literature review that was conducted on the question, can improved farm biosecurity reduce the need for antimicrobials in food animal production? Evelien, the floor is yours.
Thanks, Jeroen, for this introduction. Good morning, everyone. So we had already two nice presentations on companion animals, but as you can see, I will now make the shift to livestock production. I will present this literature search done by a colleague of us, Professor Pankaj Dhaka from India. He spent 3 months at our faculty in our team conducting this literature search. So we know antimicrobials are used in animals. Approximately 70% are consumed by the animals, and the other 30% are consumed by humans. The development of antimicrobials was life-changing for human health, but also in the livestock production, we saw that there was an increase on the animal health, the animal welfare, and the production of those animals. But of course, using antimicrobials has negative consequences, we all know, like the selection for antimicrobial resistant pathogens, with cross-resistance going together with that. And this, of course, poses a major risk for human and animal health. And the only thing that we can do to counter this is to reduce the antimicrobial usage. Antimicrobials are used in 3 ways. We know therapeutic way, of course, that's for treating diseased animals, which will remain necessary. But then there is also the prophylactic and the metaphylactic way of treating animals, which is actually not treating because you are giving antimicrobials to healthy animals, and that's of course not what we aim for. There are already large parts of the world, such as Europe, where measures are taken to reduce the use of antimicrobials by the government, legislation, the EU legislation, but there are also many other countries where this is not the case. But if we want to reduce the use of antimicrobials, we have to try to reduce the introduction and spread of pathogens in animal populations, so there are less diseased animals, so less need to use antimicrobials. And this is where biosecurity comes into the picture. But what is biosecurity? Biosecurity is defined as the set of management, behavioral, and physical measures taken to reduce the risk of introducing establishment and spread of pathogenic agents to, within, and from an animal population. And by using biosecurity measures on a farm, we can say that there is like a balance between production, food safety, animal welfare, and one health on one side, and then on the other side, there is the presence of infections, and the use of antimicrobials. When there is bad biosecurity on a farm, or the level of biosecurity on a farm is low, then there's a higher risk that there will be infections introduced into the farm and spread. This will lead to decreased production, food safety, animal welfare, and antimicrobials will be used and needed to treat those animals. While in the other case, if there is good biosecurity on a farm, higher levels of biosecurity, there is a smaller chance of introducing and spreading pathogens and infections into these animals, which will result in an increased production, food safety, animal welfare, so all positive things, and less diseased animals, so less need to use antimicrobials. But this is what we hypothesized, because the research question of this review was, is there really an association between the farm biosecurity and antimicrobials used in food-producing animals? Therefore, 3 online databases were searched, so PubMed, Scopus, and ScienceDirect, and studies were included, published between 2001 and 2022, who were written in English, published in peer-reviewed journals, and of course, they had to investigate the link between farm biosecurity and the use of antimicrobials in food-producing animals. So after searching those 3 databases with our criteria, we ended up with a little bit more than 1,000 studies. Of course, many duplicates, they were removed, so 392 were left. There were quite some papers that studied antimicrobial usage, but not biosecurity on the farm, or the other way around. So again, many papers dropped out. And then there were 63 papers left for a full text read. There were as well still some irrelevant papers for the aim of our review. So we ended up with 27 studies that were included in this literature review. All those 27 studies were carried out among 16 countries, and actually 20 of them were in Europe. There was 1 study done in Mexico, and the other 6 were Asian studies done in Japan, Vietnam, Bangladesh, and the last one I forgot, and I'm as well not so good in reading the map, so I need to take a look at it. Sorry. So as you can see, the most studies included in our review are done in Europe, while actually antimicrobial usage is as well a large problem, or large antimicrobial numbers are used in countries from low and middle income, so the countries where not a lot of data are present of. When we take a look at its species distribution, a little bit more than half of the studies were done in pig farms, then a quarter of them in poultry farms, so chickens, 3 studies done in cattle and 1 in a turkey farm, and also 2 studies that included multiple species. And 70% of the studies were cross-sectional. There were 7 longitudinal studies, of which only 4 were interventional studies, and then there was 1 case control study. If we now take a look at what those studies concluded, or what the results were, then we see that more than half of the studies found that there was a positive association between farm biosecurity, so a higher level of farm biosecurity, and reduced antimicrobial usage, and almost 20% of the studies found the same positive link between antimicrobial usage and having a good farm management. Then there were 2 studies that saw that if there was coaching and raising awareness of this problem to the farmers, that there was also a reduction in antimicrobial usage, and 1 study found that there was a positive effect on the farm economics. These studies, doing coaching and raising awareness, are quite interesting and I think very important, because we see that from dairy herds, for example, only by raising awareness to the farmer of what the benefits can be of biosecurity measures and what the risks are of using antimicrobials, they were able to reduce the antimicrobial usage with 17%. And also a study done in Belgium on pig farms by farm-specific coaching of those farms, they were able to show that it is possible to raise pigs without antibiotics and also to maintain this. So this is quite nice by doing coaching and raising awareness. Of course, we also had 5 studies which found an uncertain or spurious association between farm biosecurity and antimicrobial usage. This is probably due to some confounders, like a recent disease outbreak in those farms, like underreporting of the antimicrobials used, or just the fact that there is a lack of information on the antimicrobials used and on the level of biosecurity. So in general, these studies, actually we can say there is a positive link between having good biosecurity measures, good farm management, and less use of antimicrobials. We can go a little bit more into detail. I will go a little bit fast over this, but in general, they saw that there are some specific biosecurity measures which had a positive effect on use of antimicrobials, such as using the all-in, all-out system, using a hygiene log, using a hospital pen, proper disease management, higher cleaning and disinfection. It's important to note that I say using a hygiene log and using a hospital pen, because it's important that they use it. Just by having the hygiene log or having the hospital pen, nothing is actually going to change. So that's a thing that has to be distinguished. And then there are some other aspects, like having poor pen conditions, a high stocking density, high animal movement, or more associated with using more antimicrobials. So by doing this literature search, we also found that there are, of course, other factors affecting antimicrobial usage besides the farm biosecurity and the measurement. So with the 12 most commonly mentioned factors, there was a hypothetical causal diagram made, which you can see here, but I will explain it a little more step by step. So we see that there are factors with a direct positive effect on antimicrobial usage, which are the green stars. So if farmers have alternative to antibiotics and there is good availability of diagnostic tests, there is often less antimicrobials used on the farm. Then there are also factors with a direct negative effect on antimicrobial usage, which are the red stars. Some are logic, like if they're pushed from the pharma industry, if there is high infection rate, so many diseased animals. The economic status of the farmer plays an important role. But then you also see that intensive farming has also a direct negative effect on using antimicrobials, which might not seem so logic, but think, for example, on a dairy herd, getting more intensive, cows produce more milk, and then they are tended to get more susceptible for subclinical and clinical mastitis problems, which have to be treated with often antibiotics. So this is how using antibiotics is linked to farmers getting more intensive. And then there are some factors with a direct negative or positive effect on the use of antimicrobials, which are the black stars. Of course, the attitude of the veterinarian is important. That's something we all know. Also, the farmer's education. If he's more educated towards treating diseases, he will use more antimicrobials. If he's more educated and aware of the benefits of biosecurity and the risk of using antimicrobials, he will use less antimicrobials on his farm. And then the herd size, that's quite a complex interaction with antimicrobials, because if a herd is larger, a farmer might be more aware from, oops, if a pathogen enters my farm, yeah, I will have major losses, so I will treat sooner prophylactic, metaphylactic with antimicrobials, which is not good. But of course, you have also farmers from large herds are more aware of the fact, like, if I implement very well some biosecurity measures, I will also be able to maintain a status of not having a lot of diseased animals and using less antimicrobials. This paper is already published, so if you want to take a closer look at this diagram, you can take a look at the paper, but I will show you in the end. And then, to conclude with this review, we can make some recommendations to strengthen farm biosecurity and promote the judicious antimicrobial use. And the first thing is that the role of the animal health professional has to be reviewed, and as well working on the farmer's attitude, because implementing good biosecurity is something which involves multiple stakeholders, which requires expertise. An animal health professional, such as the veterinarian, mostly has this expertise, is in the field every day and can be the bridge between the farmer and other stakeholders like industry or the government. And we have to work on the farmer's attitude. I heard it last week on a conference. I heard it already this morning, that sometimes farmers can be quite stubborn and it's sometimes difficult to change their mind on biosecurity. And this is something that we as a veterinarian, but also other stakeholders have to influence by exchanging knowledge, by coaching, by raising awareness. But with saying that we have to teach them and knowledge have to be there, I come to the other points, but I will take them together because they are quite similar. If we want to teach them, if we want to train them, knowledge have to be there. And in Dutch we say, meten is weten. If you measure it, you know it. And that's something what is often the problem, that there are not enough data on it. So we have to measure the biosecurity, so we know the level of biosecurity on the farm. We have to measure how much antibiotics are used, and then more important, also not only in Europe, because as you can see, there are quite some studies done in Europe, but also in other parts of the world, it's important to measure biosecurity, measure antimicrobial usage, so we gain the knowledge and we can work on this. So, to conclude this presentation, biosecurity plays a vital role in the effort to reduce antimicrobial usage, but there is still some work that can be done. We have to gain more knowledge because there is still a large knowledge gap, especially in those low and middle income countries. We have to do more longitudinal studies because from those 27 studies, only 7 were longitudinal and only 4 interventional. But only with a longitudinal study, you can see if I change or I implement a biosecurity measure, what will be the effect on the use of antimicrobials, and then also better quantification of biosecurity and antimicrobial usage, not only in Europe or so, but also in those low and middle income countries. So, like I said, the paper is already published. I want to thank Pankaj for doing this nice review. If you have questions, I'll do my best to answer them, but you can also take a look at this very nice and interesting paper. Thank you very much.
Thank you, Evelien, for this interesting overview. Any questions? I don't see. There is one in the chat.
Several questions in the chat. One is coming from Geert Hoflak. He asks which biosecurity thresholds should be obtained when using the UGent biocheck for internal and external biosecurity in order to see a positive effect on antimicrobial use, and are there species differences?
That's a very nice and interesting question. To be honest, I don't know what threshold has to be reached, but of course the higher the better is something I would say. You have to aim for good biosecurity measures, the highest score, and that will have the most positive effect on your farm, I think. So I would not say be happy with 70%, no, go for the 100%, of course.
Then there's another question. Is there any reason for leaving out the US in the review?
I don't think it was left out, actually. I think there were just no papers that were fitting into our inclusion criteria found to include it. It was not on purpose left out or so.
Yeah, another question from the same person. Why less representation from Africa? Is it that the studies from Africa did not investigate both biosecurity and antimicrobial use?
Yeah, probably because we did not select on a certain part of the world or so. So everything was taken into account, but of course these studies would probably not have indeed searched both antimicrobial usage and biosecurity, and that's what we needed for this review. So that's also why I say we recommend to really implement both, look for both, and do more studies also in Africa and the other low and middle income countries.
Then I have a final question from Nancy Debergh. She asked, to convince the farmers, is it also important to look at the economic impacts? And do you also plan a study on this?
It's of course important to stress the economic impact, because I think that's in the end also the most important for the farmer, his economics. So, and I don't think we plan a study on it, but it might be of course interested to do. But indeed, if we can gain more knowledge on that and see as well, like what's the impact of implementing a biosecurity measure on the economics, on what will the farmer get from this, and as well reduce antimicrobials, I think it will have a nice overview and it will stimulate farmers to implement biosecurity measures for sure.
Okay, thank you.
Thank you, Evelien. Maybe I can just jump in on the last question. We are coordinating, and you know very good, because you do the majority of the work on the European research project Biosecure, in which we have a big work package on economics. So we definitely have a big consortium working on also the economic part of biosecurity. So we definitely will have studies on that and hope to have presentations in the future. So
we will come back to you later with this.
So with this, I want to thank you, Evelien, once more. And yep. Welcome back to the second part of this morning session. And first presentation of the second part will be given by Dr. Cécile Boland about Linezolid-resistant isolates in food-producing animals in Belgium. Cécile, the floor is yours.
Thank you, Aaron. So it's really my pleasure today to present to you the results of our study on Linezolid-resistant isolates conducted in Belgium in food-producing animals. So this work was from a research project in the framework of the One Health European Joint Programme, the Linwest project, which was co-financed by Sciensano. We have already spread the highlights of this project through different channels, but we think that it's still important to disseminate this information more widely. So that's the reason why I will present it again today to this audience, and yeah, thank you for the opportunity. So linezolid is an antibiotic that is critically important to treat human infections caused, among others, by methicillin-resistant Staphylococcus aureus and vancomycin-resistant Enterococci. This antimicrobial is not licensed in food-producing animals, so it's used in human medicine. However, prior to this study, some linezolid-resistant isolates were reported in some European countries in both humans and animals. These are different resistant mechanisms that are known to confer resistance to linezolid. First of all, these are mutations in the 23S ribosomal RNA. These are, of course, non-transferable. But besides to this, 3 genes are known to be transferable. These are CF3, OPTRA, and POXTA. These genes encode resistance to linezolid, belonging to the oxazolidinone class. But not only. These are also other resistance, in particular to antimicrobial, used commonly or very commonly in food-producing animals. So these are written in green on these slides. These are phenycols, lancosamide, chloromethylene, and tetracycline. So these 3 genes confer the resistance to these other antibiotics. One unique common point to these 3 genes is the common cross-resistance to phenycols. So keep it in mind, this information for the following, it's important. So this raised the questions whether a cross-selection of linezolid resistance could occur through the use of non-critical antibiotics. The aims of our study were to investigate the linezolid resistance occurrence in food-producing animals in Belgium, and to assess this occurrence through selective monitoring, to analyze the determinants involved in this resistance to answer the questions whether or not there could be transferred, and whether a cross-selection could occur, to study what is surrounding the linezolid-resistant genes in the collected isolates, and the genetic relatedness between the different isolates, including human isolates that were previously collected in Belgium. So we based on study on the samples that were collected for the official antimicrobial resistance monitoring from healthy animals. So these are nasal swabs from pigs. In 2019, it was from pigs, dedicated initially for the monitoring of MRSA, but other bacteria are able to grow on the same medium. and fecal samples from different animal categories taken for the monitoring of enterococci. So after having conducted the official monitoring, we gather all the bacteria grown on the petri dishes and spread them all on a selective medium. So this was Columbia sheep blood agar, supplemented with linezolid at a threshold concentration, 4 milligram per liter. We incubated the plates during 48 hours. And after that, if we observe different morphologies of colony, we selected up to 2 different morphologies and identified the bacterial species through MALDI-TOF mass spectrometry. So this was important to make first distinction between gram-negative and gram-positive bacteria, because gram-negative bacteria are intrinsically resistant to linezolid. So these were not kept for the study. We were only focused on the gram-positive one, and especially Staphylococcus and Enterococci. Afterwards, we established the minimum inhibitory concentration of linezolid and of other antimicrobials by Bro's microdeletion with Sanzi-Titer plates. And we compared the incubation between 24 hours, which is the recommended reading time, and 48 hours of incubation before the reading. And finally, we use the whole genome sequencing to answer the different research questions. Let's now have a look at our results. So first, the assessment of the linezolid-resistant occurrence. So the criteria was we consider as positive for linezolid-resistant occurrence the sample for which we observed bacterial growth on the medium supplemented with linezolid and with gram-positive bacteria grown on this plate. So we used fecal samples and nasal swab samples, as explained before. And you can see here that we observed different occurrence of linezolid-resistant bacteria depending on the animal categories. So the lower rates were observed in poultry, none were observed in turkey, and up to 6% in breeding hens. And the higher rate were observed in veal calves and pigs. So in veal calves, we observed 16% of the samples that carried linezolid-resistant bacteria. And in pigs, we observe different percentage depending on the kind of samples that we analyze. But I have to say that it was not collected from the same animals, so we cannot directly compare the site of sampling. But yeah, whatever, we observed 11% in fecal samples from pigs and up to 25% in nasal swabs from fattening pigs. So this means 1 out of 4 samples from fattening pigs carried at least 1 linezolid-resistant bacteria. So this was surprising high. And another surprise that came from our study was that we found several linezolid-resistant enterococci in nasal swabs. While until now, the nasal swabs are only used for the monitoring of MRSA in animals, and not for enterococci, the monitoring is only conducted on fecal samples. So this is certainly a matrix interesting to study for enterococci also. Let's now have a look at the genetic determinants. So we sequenced by whole genome sequencing 147 isolates. These are the isolates collected from food-producing animals and a few from humans that were collected in Belgium. Most of the isolates, 97%, carried linezolid-resistant genes known to be transferable. And so you can see it in yellow on the circle. And if we look deeper in these isolates, you can see that in blue, most of the isolates carry 1 resistant gene to linezolid, but some isolates in gray, 18 carried 2 different genes encoding linezolid resistance, and even in one isolate, we found the 3 genes, CFR, OPTRA, and POXTA. So now what about the repartition of the genes? So we found mainly OPTRA in the collection of isolates, And it's important because these genes encode specifically only a cross-resistance to phenicals beside the linezolid resistance. We observe various genetic environments surrounding the linezolid-resistant genes, with genes encoding for resistance to other antimicrobials, phenical, macrolides, and aminoglycoside. So there is a genetic diversity of the environment of the linezolid-resistant genes. Also, interesting finding in more than 95% of the isolates, so almost all, carried also other phenical-resistant genes, defects E and defects B, in addition to the linezolid-resistant genes. And the second gene that was found the most frequently was POX-A, which could also be selected through the use of tetracyclines, in addition to phenicals. So we conducted afterwards a large phylogenomic analysis, and we observed that we observed very large phylogenetic differences with some smaller clades containing more closely related isolates. So among our collection, we observe 28 different sequence types in Enterococcus faecalis isolates and 32 different sequence types in Enterococcus faecium. So it was diverse. When looking at some cluster, we saw that even in a lineage, a single lineage, the sequence type 480, that is known to be quite frequent in clinical settings. We observed this in isolates from different hosts, human, bovine, and pigs. And we observed this in different locations in Belgium. And we observed in these smaller clades, we observed different alleles of linezolid-resistant genes. So globally, we did not observe a clear correlation between the phylogenies and the alleles of linezolid-resistant genes. And so it could indicate a facilitated transfer and spread of linezolid-resistant genes. So the key word on this complicated slide is genetic diversity. Now, besides this global diversity, we observe some clades with closely related isolates. So here, highlighted in orange, you can see 9 isolates from different hosts, bovine, pigs, and humans. And these differ only by 10 up to 25 SNPs. So on the entire genome, it's only a few differences. And so this closeness between isolates from different hosts suggests a potential epidemiological link. So let's now move to the several take-home messages that you keep in mind afterwards. So linezolid resistance is critical for human health and is already well established within the animal bacteria. The highest linezolid resistant rates were observed in veal calves, 16%, and pigs up to 25%. We observed in most of the isolates transferable linezolid resistant genes. Our results suggest a putative link with the use of fluoroquinolone, given that more than 95% of the isolates carried other phenical-resistant genes, and that the 3 genes CFR, OPTR, and POX-A found give cross-resistance to linezolid and phenical. So such cross-selection will maintain this gene. We observe large phylogenomic distances between clades of more related isolates. So in other words, we would only ever observe the tip of the iceberg, and a largely uncharacterized reservoir of linezolid-resistant isolates would be circulating. We observed genomic similarity between Enterococcus faecalis isolates collected from various host species, including humans. So this suggests that a spillover of such strain could occur. And also, besides to this threat, another threat is the risk of air-mare transmission to humans. So through our selective monitoring approach, we observed a much broader reservoir of linezolid-resistant bacteria. So it sounds quite logical if you look with selective pressure with antimicrobials, but the level of occurrence was quite unexpected. So we would recommend to conduct such selective monitoring, to continue this monitoring, and to include whole genome sequencing to follow the different lineages of bacteria carrying this resistance, and with a 48 hours of incubation. And also the addition of nasal sampling for enterococci monitoring could be of added value. So what are the consequences of our study and of our findings? So the apparition of linezolid-resistant bacteria is a problem because linezolid is a last resort antibiotic for human health. Linezolid-resistant genes give resistance to other antibiotics, mainly phenicals, and the cross-selection will maintain these genes, and new antibiotics are rare. So our results highlight the need to promote the prudent use of all antibiotics, not only the critical ones for human health, but really all the antibiotics in a One Health perspective. And now, what's next after this work? We are now working with the European Reference Laboratory for Antimicrobial Resistance to try to set up an harmonized monitoring with other member states to assess the situation in the other European countries. It is on voluntary basis, and yeah, there is an interest from the other member states. And in Belgium, based on our studies and on also other studies in the literature, the AMCRA decided to launch a working group to discuss about the guidelines for the use of fluoroquinolone in animals. And the work is currently in progress, and the conclusion will be published in an advice in the coming months. So if you want more details, you may have a look at our publication. It's in open access, and you have here the references. I want to thank you all for your attention, and a special thank to Michael Timmermans, who is a PhD student that conducted this research project. And thank you to all the people in my lab, but also the other colleagues and partners that contributed to this study. Thank you.
Thank you very much, Cécile. Are there questions? Can you maybe just wait for the microphone, also for the people at home to be able to hear your...
Did you look at...
yeah, it's on.
Did you look at other species within the bovines, but veal calves?
Not through this monitoring. It was only in veal calves for the moment. But we will have a look in the future in other category from the nasal samples. For the samples taken for the monitoring of enterococci, it is only veal calves, so we don't have access to the samples. But for the MRSA, we are also looking at dairy cattle and young beef cattle.
Yes, thank you for this interesting talk. Are there any data available about the prevalence of resistance to linezolid in enterococci of human origin? We got here the data on animals.
Yes, we look at this with my colleague for the human epidemiological team. For the moment, there are some, and the awareness is raising, so the NRC of enterococci is receiving more because they raise their awareness. So it's difficult to have an actual prevalence because the NRC is collecting the samples, but it's not a randomized study, or But yeah, it seems that there are some. We don't know yet if it's increasing or not. For the moment, it's still under control, but it's really important to avoid that it increases and to avoid the spread of that. Yeah.
Okay, thank you.
No more additional questions, not from the chat either. Well, thank you once again, Cécile, for this nice presentation. And the next speaker is Moniek Ringenier from Gent University, and Moniek will present on the topic where she is also making her PhD on, and that is Fluoroquinolone resistance in E. coli in the broiler production chain and specifically the role of the parent stock and the hatchery. The floor is yours.
Thank you. Good morning, everyone. I'm happy to share with you the results of our research, as mentioned, on fluoroquinolone resistance in E. coli in the broiler production chain, specifically also on the role of parent stock and the hatchery. But first, I want to take you into the story of the why. Why did we do this research? And it started already quite a few years ago in a cross-border project together with the Netherlands, where we looked into ESBL producing ertapenem and ciprofloxacin-resistant E. coli in broiler farms, both in the Netherlands and in Belgium. What did we do? Well, we sampled broiler farms at the end of the production cycle, and we took feces samples. And here we see a small part of the results for the broiler farm. So on the left, you see the graph for Belgium. On the right, the graph for the Netherlands. You see for Belgium, we sampled 15 farms, so every number is corresponding to a farm. And in the Netherlands, we sampled 14 farms. And on the y-axis, you see the percentage of samples that were positive, the white bars being the ESBL producing E. coli, and the gray bars being the ciprofloxacin resistant E. coli. And as you can see in both countries, we saw a high number of positive samples for ciprofloxacin resistant E. coli. And we also see that in Belgium, the numbers were higher than in the Netherlands. We also collected a lot of data on antimicrobial usage of the specific production round we sampled, but also of the year before. And if we looked into the historic use, we saw that the usage of fluoroquinolones was higher in the Netherlands than in Belgium, which was also a bit surprising when we see these results. So with this in mind, we came up with a new research project to look at where does it come from here in Belgium. Where does it come from? How does it enter these broiler farms? So what we first did was a cross-sectional study, purely on broiler farms, where we sampled 29 farms. Each farm we visited 4 times. At day 0, we visited the farm and we took samples from the environment before the day-old chicks entered. The stable was already ready for the arrival of the chicks, and we also sampled 30 chicks before they entered the stable to see if, do they come in on the farm with the chicks? Is it already in the environment? What plays a role? And then again, we came back 3 more times to the same farm to sample the same flock of animals to see is there maybe another moment, like let's say it doesn't come in with the day-old chicks or in the environment, is there another point in time through the cycle where they appear, and how do they spread within the same flock of animals? And in the end, at day 35, we also took feather samples to look at the presence of antimicrobial residues, specifically fluoroquinolones, as an extra validation to see that they really didn't use fluoroquinolones. So just as where does it come from? We did whole genome sequencing on isolates from the study. What did we find? Well, we found already in the day-old chicks, before they entered the farm, we already found fluoroquinolone resistant E. coli. But we also found it in the environment. So in both parts, they were already present. And then again, according to core genome MLST, we found that isolates that were present in the beginning of the production round stayed present until the end. So at day 35, they were identical isolates without the farms using any fluoroquinolones. So this also shows that the isolates that are already present in the beginning stay on the farm during the whole production cycle. So knowing this, we know, okay, the environment, important role is cleaning and disinfection. But what about these day-old chicks? How do they get, where do those fluoroquinolone resistant E. coli come from? What is the role of the broiler production chain? Because we know these day-old chicks, they come from parent stock farms. It's a whole, the production pyramid. You have the parent stock farms, they lay the eggs go to the hatchery, and then from the hatchery, the day-old chicks go to the broiler farms. On parent stock farms, antimicrobial usage plays also a role. And you wonder, okay, in the hatchery, do we find these fluoroquinolone resistant E. coli already in the environment of the hatchery? So we did a longitudinal study in which we included 4 parent stock farms. On those parent stock farms, when we visited that day, we followed the batch of eggs that was laid on that day, and that batch of eggs we followed to the hatchery, and we followed the whole batch through the whole process of the hatchery until day of hatching. What did we collect? So on the parent stock farm data, we collected data on farm characteristics, antimicrobial usage, and on biosecurity using the biocheck.ugent. And samples, we collected a lot of samples. So first, just to give you an idea what we did with the samples. So all the samples we collected, so both environmental and animal samples, we plated them on McConkey plates to look for sensitive E. coli, and also on McConkey plates enriched with enrofloxacin to see if these E. coli were also resistant to fluoroquinolones. So what did we do? What did we sample on the parent stock farm? So first we selected one poultry house randomly, and in that house we collected 30 swabs of fecal droppings. We collected 30 eggs coming from that specific house. And those eggs, we didn't just look at the outside of the eggs, but we looked into the outside, but also the eggshell and the egg pores, and in the inside of the eggs for the presence of fluoroquinolone-resistant E. coli. And also we took environmental swabs. So on the parent stock farm, we took a swab from the egg tray. It's the image you can. Oh, can I go back? Yeah, it's the image you see below. That's the egg tray. The egg trays are being delivered from the hatchery to the parent stock farms, and on those egg trays, they collect the eggs, and they will also go to the hatchery on those eggs and are being incubated on those trays. And again, on these farms, we also took feather samples to look at the presence of fluoroquinolone residues. In the hatchery, we visited the hatchery 2 times. So we selected those moments in the hatcheries which were important for a possible transfer of the fluoroquinolone resistance E. coli. So we took day 18, around day 18, it's the day that the eggs are being transferred. So the eggs could first go into the incubator, and once around day 18, they are being transferred with these suction cups, as you can see on the right of the screen, into hatching trays. So first they are in the egg tray, the picture I showed before, which you can see also right above on the screen. And with the help of the suction cups, they are transferred into the hatching trays, which are the white trays, which you can see over there. So we also took swabs of those locations. And then again, we came back a day of hatching. So then, the eggs are being hatched. They are being in those trays, just as I showed before. They are going onto the collection belt. And on the left, we took swabs of 2 locations of the collection belt. One location was where the eggs were separated from the day-old chicks. So they emptied the trays, and the eggs and chicks are separated. So there we took a swab, and then we took a swab again at the point of the belt where they selected the good and the bad chicks. And we took samples again from the trays in which the chickens are then collected to go to the farm. And also we took 30 day-old chicks at the end of this process to see if they had fluoroquinolone-resistant E. coli in their intestines. So now the results. What did we get it for information about antimicrobial usage? Here on the left column you see the farm ID. There were in the end 4 farms, and in the second column you see the age of the parent stock. So they were relatively young animals. And then on the third column you see the antimicrobial usage on the rearing farm, and in the last column the antimicrobial usage on the parent stock farm. So the parent stock, they first are until like 17, 20 weeks of age. They stay on a rearing farm, and you see that on 3 out of 4 rearing farms antimicrobials were used. And in the parent stock farm, only 1 farm used antimicrobials. But as you can see, none of them were fluoroquinolones. Then into the results for the fluoroquinolone-resistant E. coli. In the parent stock farm, here we see a graph where you see on the x-axis you see the 4 different farms we sampled. On the y-axis you see the number of samples we took. So there were every time 30. And then the blue bar indicates if we found E. coli, and the orange bar indicates if it were fluoroquinolone resistant E. coli. Yeah, it doesn't surprise us that we find E. coli in feces samples from chickens, but as we do see is that a lot of them were also fluoroquinolone resistant, without the parent stock using any fluoroquinolones. Then if we look into the eggs, so these are the eggs that were collected at the parent stock farm, just after laying. And here we see, so again, the 4 farms on the x-axis, the number of samples on the y-axis, and here we could only find in a low number of samples E. coli, but none of them were fluoroquinolone resistant. Then we go to the hatchery. In the hatchery we collected the eggs at day 18, and here we again see the same image as the eggs on the parent stock farm. We could find on a few samples E. coli, but none of them resistant. And then the day-old chicks. We already find, although the animals are not old, they're just a few hours old, some maybe a bit older than the others, but we again find E. coli, and also in some batches quite a lot of them were fluoroquinolone resistant. So this gives us an image of the animal samples we collected. You already see, okay, they are present in the parent stock, they are there in the day-old chicks. Okay, in the eggs, we cannot really find them. So what kind of role does the eggs play in the dissemination of fluoroquinolone-resistant E. coli? But we took also environmental samples. Here we see, on the x-axis, the different locations we sampled. So from the left is the egg tray, then you have the hatching tray, then the suction cups, then the 2 different locations of the collection belt where the day-old chicks go over, and then at the end you see the trays, the transport trays of the chicks. And on the y-axis you see the number of hatcheries positive. And here I only show you the fluoroquinolone resistant E. coli. And what you see is, okay, the egg trays that were sampled on the parent stock farms, we didn't find any. On the transport of the hatching trays, also none. But we did find them on the suction cups, and we did find them on all hatcheries at the collection belt. So the collection belt is where all chicks go over before they go to the broiler farm. So this was quite a lot in all 4 hatcheries. And in the transport on the transport trays, we only found it in 2 out of 4 hatcheries. So going back again to the question, what is the role of the broiler production chain? As already mentioned, they are there in the parent stock. We do find them in the environment of the hatchery, and also again in the day-old chicks, even though they are really young, we also find them here. We knew we found them when they arrived on the farm, but we already can find them in the chicks at the hatchery. We do know, or our research show, that eggs are probably not an important route for dissemination, but it shows us that the environment of the hatchery is a critical point for the spread to these day-old chicks. So this is also so for future research, it will be interesting to see what could be a possible role of on-farm hatching. Because on on-farm hatching, you don't have this part where all the chicks go over the collection belt, but the eggs are being transferred immediately to the broiler farm itself, and would that maybe be a possible solution to reduce the prevalence of this fluoroquinolone resistance E. coli in broilers? Thank you.
Thank you, Monique, for this interesting results. Are there questions? There's a few in the left side there. The micro is coming.
Thank you very much for the presentation. So it's clear that the environment is the problem. I don't know if you have checked it for those isolates. In the environment, if you check the resistance to biocides, because maybe the reason is that they are persistent and they are not eliminated during the disinfection?
For the isolates of this specific longitudinal study, we are going to perform whole genome sequencing. We already did we also looked for the biosite genes in the isolates from the cross-sectional study, but here so far we couldn't find any correlation with the resistance against the biosites. But it should be mentioned though that the isolates we found in the cross-sectional study were isolates that were resistant against a lot of antibiotics, so often against more than 3 groups of antibiotics. So this also shows, like, we know on broiler farms or parent stock farms, antimicrobials are being used. So also by using other antibiotics, you can also select for fluoroquinolone resistance. So yeah.
Maybe it was told in your presentation, but I'm not sure. Do you look inside the eggs or only on the shells?
We looked, at the outside of the egg. So we first kind of extracted the bacteria from the outside, then also the pores of the egg. And inside the membrane, and then also indeed the content. But on day 18, it must be mentioned, then it's already an embryo. So we had the embryo, and from the embryo we took the intestines as well. And there we couldn't find any, and also not on the parent stock farm, where it's still egg yolk, and we also didn't find any. But I mean, it's possible that we didn't find it, but it was there, but that our method was not sensitive enough, that I cannot tell. But so far, we couldn't find them.
Okay, thank you.
The question in the back, Wannes.
Thank you, Moniek, for the very nice presentation and results. Those 4 hatcheries, were they all Belgian or Belgian and Dutch?
This study, we only focused on Belgium, yeah.
Okay. And I wondered, so is there like something like a hatchery, or were there quite big differences in the management of those hatcheries in terms of cleaning, for example, or is it all really comparable? And is there anything in those hatcheries that would explain the results that you find?
I find it difficult to. We did collect information about the cleaning and disinfection of the hatchery. So on paper, it is similar in all 4. But then again, yeah, you have what you have on paper and how you do it in real life. There were differences in how they applied their protocols. They were there, yeah. I don't want to pinpoint on something specific.
No, that was not the question. Thank you.
Yeah.
Question from someone at home, Ludwig Hendricks. Since no fluoroquinolones were used in the parent stock, do you have a theory on where fluoroquinolone resistance comes from originally?
Yeah. What we did find in the broiler farms was, as I said, the multi-drug resistance. So by using, if it's there and you use other antibiotics, they will stay in the environment or in the animals and you will not get rid of it. We did see though that there was a farm that had a really big salmonella issue and they did a really good cleaning and disinfection protocol, but it was really intense and then you couldn't find anything in your environment. So once you are maybe rid of those isolates and you don't use antibiotics, but that's a really ideal, so I don't know if it's, and where it comes from, yeah, it's historic use as well.
And then a question from Nancy De Bruin. Do you have any specific recommendations for hatcheries to prevent environmental contamination or on the parent stock selection?
Yeah, the thing is with the hatcheries, the batches, they go over the let's say the collection belt, they run over the belt batch by batch without any cleaning and disinfection in between, because that makes their work impossible because it's so large number of animals. But that makes it also really difficult. Let's say if you have a few animals that do have these resistant E. coli with them, go over the belt. that's why I thought it would be interesting to look at on-farm hatching as a possible solution, because I think in let's say cleaning disinfection between batches of animals, it makes it really difficult. Yeah.
No more questions. Well, thank you once again, Monique, for this very interesting presentation. And that brings us already to the last presentation of this morning session. And this presentation will be brought to us by Léonard Théron from Rumen Expert. And the title of the presentation is Prevention of Calf Respiratory Pathologies Using a New Herbal Supplement, a Preliminary Study. Léonard, the floor is yours.
Hi everybody at home. This is a small preliminary study that is a part of 6 different parallel studies in young calf. In fact, for those who don't know Rumen Expert, we are a small animal health CRO, so basically we make happen trials on the field in all the necessary conditions. This study was aiming to find alternatives to potential treatments or reduction of use of antibiotics using a specific veterinary company that is using mostly mixture of herbal components. The given mixture is including some fungi, some plants, some roots, which are available online if you need to check. But the formal composition is still industrial secret, of course. The primary objective of the study was to try to reduce and prevent 50% of neonatal disease, mostly focusing on respiratory disease and digestive disease, with a statistical power of 80% and 90% confidence interval. And if not reached, the secondary objective would be to have a specific look on the days of antibiotherapy, the days under disease, and some lesion score depending on the disease you're looking at. It was set as a multicentric, double-blind, placebo-controlled study with 2 different phytocomplex tested, 2 different composition. Basically, the setup was to have all groups in all of the centers of experimentation. So it was decided to go for 4 farms and have 3 groups in all farms. The ethical protocol was validated by Liège Commission, and the inclusion criteria was quite normal for these kind of trials. We tried to set the initial epidemiological settings to have 120 calf in this study. The 2 phytocomplexes are slightly different. One is more, let's say, action on the immunity itself, and the second one more on the digestive tract physiology. And the placebo was basically the remaining of the 2 phytocomplex, only the non-active components. How we did this? We included the calf from birth, and we followed them until 6 months for barometric measurements. So all the calves were weighted at birth. Then under 2 to 7 days, they begin to receive these phytocomplex for 10 days. And during that period, they were also evaluated for their cholesterol transfer. They got some specific metabolic profiles. And we did some microbiological analysis of feces. If there was an onset of disease, they had extra PCR also. And they were also, all the treatments and mandatory, action managements were noted. At 20 to 28 days, they got specific metabolic profiles, they got specific ultrasounds on the lungs, full hematology, checking for a lot of specific from the bovine inflammatory factors, and also reanalysis of the microbiology of the feces. And then at weaning, we did about the same, but keeping mostly the measurement of the biomarkers. What was the goal? The goal was to identify a potential prevention and then to check on the antibiotic alternative. Out of 120 calves recruited, 114 animals make it through the study in the 4 farms. And if you look at the distribution of the animals through the full farm, you will have on the left side, so in my graph, the red bars, this is the phytocomplex 1. The green one is the control group, and the blue lines is the phytocomplex 2. The sex ratio was even, the weight at birth was even, the distribution between farms was even, and one thing that was really interesting is that the weight at birth was really high. So most of the people, when they talk about Belgian blue breeds, they talk about calves between 46 and 49 kilos. But on all the studies that we've been doing on the last years, we've seen an increase of the weight of these calves and probably also the requirements of these calves. So there's also questions to be open on that. If we look specifically at the 3 groups when it comes to the transfer of antibodies from the colostrum, this is even, so there is no difference between the groups, between the phytocomplex 1, the placebo group, and the phytocomplex 2. It's the same for the total protein, no significant difference, and also for the albumin. There's 10 more biomarkers that we tested, but at the start of the study, all the cows were comparable. I invite you to check that the Brix level in IgG of the first week was quite high, so 9.3 is equivalent to 22 grams of IgG per liter of blood. So it means that on average it was calf that receive a nice transfer of immunity. This is quite non-coherent with the classical field of trials, because classically you would expect to have 8.7, 8.5 average. Did we succeed in the first objective, the principal objective, to decrease 50% of neonatal pathologies onset? The answer is no. So at that stage, there was no significant difference between the 3 groups, though there was a tendency to have a diminishing in the phytocomplex 2 group compared to the placebo group. On average, 60% of the animals had a disease, which is either a diarrhea or a pneumonia. Diarrhea was present in 65% of the animals, and pneumonia happened in 25% of the animals. If we look at the survival curve of disease, when we include all the animals in that trial, in the phytocomplex 2 group, there was a significant reduction of 25%, a tendency of reduction of 25% at that stage with the dosage used and the trial settings. If we look in the number of treatment days, so to be clear, the animals, whatever groups they were in, had just a mandatory condition in the beginning of the trial. They had 1 day under NSAID before onset of antibiotic treatments to reduce the habit of the farmer to go directly for antibiotics on normal happening disease. So the bias that we induced in that trial was that we just asked them that whatever groups it was, because it was double blind, they go for 1 day of NSAID before onsetting any antibiotics. The number of treatment days, whatever disease we looked at, there was a reduction of 30%, but non-significant, specifically for the phytocomplex 2 group compared to the placebo. And if we look specifically of inside the treatment, there was an early significant reduction in the antibiotics, so the p-value was 0.07, but there was a global minus 45% reduction in numbers of days of treatments under antibiotics using the phytocomplex 2. If we look at the grade of pathology for specifically the respiratory calf, there was a reduction, but non-significant, in the severity of the disease in the phytocomplex 2 group, which was not observed in the phytocomplex 1 group. And there was also a decrease in lung parenchyma lesions by ultrasound after 10 or 15 days after disease in the phytocomplex 2 group. When we observe all the biomarkers from this trial, so specifically all the biomarkers in the range of inflammation, there was no significant difference in the beginning or at weaning. There was no real difference between the calf. It was the same for the oxidative stress biomarkers, so we tried to be as large as possible because the producer of the phytocomplex was not exactly sure of the way of action of the complex. And we checked, glutathione peroxidase, MDA, TAS, oxidative capacity in the animals, and there was some tendency, but no significant differences. There was one tendency that was interesting, is in the biomarkers from the nutrition. There was, in the phytocomplex 2, there was a tendency to have an increased albumin level around the study, which could indicate subclinical capacity to recover that was slightly improved by the use of the phytocomplex. When we compare the productivity of these calves, so average daily gain at weaning at 6 months, there was no significant difference from the group. And if you look at from a continuous perspective, you can see that the data has some discrepancy, which is the complexity of doing field studies is always that this is not a control condition. So voilà. What was the main summary of that one study, because it's a field of 6 different studies, is that at the dosage that we use in this trial, there was a significant difference between the 2 phytocomplex, but the difference between the placebo and the phytocomplex 2 was slightly too low to be shown significant. Main reason is that the hypothesis that we set in the beginning, 50% reduction of disease, was probably too high, because we've seen a reduction of 25%, but the setting, 120 calf, is not able to show a significant difference of 25. What was really interesting is that there was a clean and strong tendency for antibiotic reduction using the phytocomplex 2 of 45% and 10% long-term ultrasound lesions. What was also interesting in that study is that we've seen that some calves that were exposed to the phytocomplex 2 had a stronger nutrition recuperation, which was also a tendency showing that, when treatments are lower, probably the calf are going back on track faster. What we can say from that? We can say that in the field of alternative to antibiotics, there is no much, okay? So if you look at the market for now, we are looking a lot at the resistance in the bugs. We are looking a lot at control measure to how to use the antibiotics. But the financement of innovation in the field of alternative of antibiotics is still a bit low. What we commit as a CRO is try to only do trials for alternative to antibiotics. And the first one of a medium-sized trial was quite convincing to go further. So now the new steps is changing the dosage and enlarging the size of the study. Thank you very much.
Thank you, Leonard, for this interesting presentation and showing how difficult it is to do this type of studies in the field. Any questions? Questions online? Question from my side, maybe you said it, but are these products already commercially available or not? They're still in the development phase, or?
They are available in companion animals.
Okay.
But for different reasons, some of the components from the companion animals are not available in production animals for legal reasons. So we had a reformulation adapted to the ruminants, and we asked, of course, for derogation for the use in animals, because at least spirulina is not yet completely authorized fully authorized in ruminants. So it was necessary to have a derogation to use this. The aim of these products is both on the nutrition side, so it gives them a lot of interesting amino acids to the calf. So there's an objective to get back to the quality of the nutrition. But most of the existing components are actually close to effect that we know about ginseng, about echinacea, and stuff like that.
Yeah. Wow. Thank you. If there are no more questions, I think I can thank you once more for the presentation. And with this, I also want to thank all the presenters of this morning, especially our invited speaker, Dr. Els Broens, but all the other presentations. I can talk for myself. I found it extremely interesting, and I hope you also did so. So maybe we give them another round of applause for all the presenters this morning. Before you run off, so a few more practical things. As I mentioned in the beginning, the whole session was recorded. Soon this recording will become available on the AMCA website. So if you want to look back at some presentations, you can do it there. Also, the posters are still out there in the hallway and also on the website. Those of you that have registered for lunch, we're now going to lunch, and at 1 o'clock we start again, and I can all warmly invite you to stay here, to stay for the afternoon session, because I can promise you we have some exciting things to talk about in the afternoon session. So please, I hope to see you all back at 1 o'clock. Thank you.